Hello guys, today I will discuss the DNA replication. As you know that what is the DNA replication? DNA replication is the duplication of the DNA. Exact copy of the DNA will be formed from a parent DNA is called DNA replication. So now I don't discussed the DNA replication in details, but I will discuss the proteins, how many proteins involved in the DNA replication of prokaryotes, because protein is the most important to perform the function of DNA replication. Without protein, DNA replication unable to proceed. So let's start. First, initiation. Initiation means starting. Proteins DNA. DNA will be activated if he have ATP. DNA help in the bending of DNA. It bind to the 9-mer position of the DNA and which bended to each other. For example, is the DNA. If a DNA protein activate by ATP, it bind to here and this DNA will be bend. If DNA protein is replaced by the ADP, then he is inactive. DNA act as helicase. As you know that what is the helicase? Helicase is a hexamer proteins. It bind to the DNA and holding two strand of the DNA. For example, this is the DNA 5' to 3', 3' to 5', and these helicase hexamer circle, hexamer circle is bind to here and create a bulb. Both the strand of the DNA will 3' to 5' holded to each other, and here binded at, and it act as the recruitment of primers here and here. Single stranded binding protein. Single stranded binding protein holding the two strands of the DNA and prevent the re-annealing of the DNA. Like as this is the DNA, he convert into the follicle form, and this proteins, a single stranded protein bind to here, single stranded binding protein, and prevent these two strands converted into like that. Both the strands of the DNA is holding to each other and prevent to re-annealing to each other. So single binding proteins help in that. DNA polymerase third is a main enzyme of the DNA replications because he's at the nucleotide in 3' region of the DNA and extended to 5 towards the 5' region, and the structure of the DNA polymerase third have following subunits alpha, epsilon, theta help in the core polymerase, and psi, delta, gamma help in beta clamp loading. These subunits together is called a sliding clamp, holding the DNA polymerase if during the adding the nucleotide. DNA polymerase 1 is a help in the DNA repair and also add the nucleotide during the filling the gap, like as this nucleotide will removed from that. Here DNA polymerase recruitment a particular nucleotide to fill the gap, and DNA ligase help in these nucleotide between two nucleotides sealing the gap to each other, and finally this DNA will converted like that. And exonucleases help in the RNA primer removal. Is remove the RNA primers. What is the primer? Primer genes DNA G act as the primase. Primase help in the recruitment of nucleotide 5' to 3'. Recruitment of nucleotide upon the 3' region of the DNA, because here OH group available and here 3-phosphate group will be available. So 5 or 6 nucleotide added here, and gradually DNA polymerase third coming, which also proceed towards the 5' direction. So in next video I will also discuss the proteins involved in DNA replication of eukaryotes. Thank you.